Hello students, I'm from Meso Study and in this session we are going to discuss about the important question from the NEET Biology and the chapter name was Biological Classification. So let's start the session and in this session we take the question one by one. The first question is the phylogenetic system refers to grouping according to floral similarities, and option B includes grouping of plant in order to their increasing complexities. Option C is grouping according to evolutionary trends, and option D is grouping according to all morphological characters. To better understand, first of all, we discuss about what is phylogenetic system. The phylogenetic system is also known as cladatics. Cladatics. As we all know, these phylogenetic system and cladatics is based on evolutionary sequence as well as the evolutionary sequence plus genetic relationships among organisms. These phylogenetic system work on evolutionary sequence and the genetic relationships of organism. After giving the various theories and the equation, the first or the better understanding and accepted by the Engler and Prandtl system by the scientist Engler and Prandtl system of classification, due to which, and that classification is given in year 1892. According to the classification, phylogenetic system is the most accepted and that generally work on evolutionary and the genetic relationship of organisms. The first phylogenetic system of classification, first phylogenetic system of classification includes all plants generally from algae, from algae to angiosperms. And that generally arranged in the evolutionary sequence. And that generally evolutionary sequence is based on their complexity levels. That is generally from the simplicity, more simple to the most complex evolutionary sequence. So the right option for this is grouping include. Option includes grouping includes in phylogenetic grouping generally includes in phylogenetic grouping generally includes under the evolutionary aspects. So the right answer is the evolutionary sequence and evolutionary trends. So after discussing the question, the right answer for this is a grouping according to the evolutionary trends. So moving to the next question. The next question is, in which kingdom would you classify the archaea and nitrogen fixing organism if the five kingdom system of classification is used? As we all know, five kingdom classification was given by the R.H. Whittaker, And first of all, discussing, we generally go through the options. Option A includes Plantae, B includes Fungi, C includes Protista, and D is Monera. So better understanding for the question, as we all know, five kingdom classification was given by the scientist named as R.H. Whittaker. And he proposed the five kingdom classification in 1969. According to the concept, and he generally based the five kingdom classification on basis of, the parameters are on basis of, first one is complexity of cell structure. First is complexity of cell structure. The next one is body organization. The next one is mode of nutrition. R.H. Whittaker generally classify the various organisms in five kingdoms on the basis of these 3 points, that is complexity of cell structure, body organization, and mode of nutrition. As we all know, first they are going to discuss about the first is Monera. As we all know, later in the six kingdom classification, Monera is divided again into the two types, Archaebacteria and the Eubacteria. Firstly, in five kingdom classification, Monera includes all the prokaryotes that are generally unicellular, with peptidoglycan in their cell wall. This is the unique feature that generally possessed by the monarids. Along with that, they generally includes naked circular DNA. It means the DNA and the nuclear material are not bounded in the cell membranes. So cell bounded organelles are absent in the monarids. Along with that, the ribosomes are present only of 70S type. As we all know, ribosomes are of two types, 70S and the 80S, and S is word referred for the Sweden unit. And along with that, they generally, the mode of nutrition in monarids are heterotrophic, phototrophic, and the last one is chemotrophic. The bacteria are generally known as the various type on the basis of mode of activity. First is the blue-green algae, and also known as the cyanobacteria, nitrogen-fixing bacteria, and methanogenic archaebacteria. So these all features are come under only one kingdom known as Kingdom Monera. So the right answer for the question is Kingdom Monera, that generally categorized into the Archaeobacteria and Eubacteria in six kingdom classification given by the Carl Woese. So in five kingdom, the right answer for the question is kingdom Monera, option D. Let's move on to the next question. The next question is maximum modes of nutrition are found in. The options are option A is Monera, B includes Protista, C fungi, and D is plantae. To better understand about the topic, as we all know, mode of nutrition is one of the major aspect of the categorization of the various kingdom. As we discussed in the prior question, in kingdom Monera there are various kinds of mode of nutrition that generally may be the heterotrophic. Heterotrophic includes saprophytes, parasites, and symbionts, etc. These all are come under the mode heterotrophic. That this was the first one. The next, they are some kind of monerans are autotrophic as well. It means they are self-capable of producing their own food by themselves. In autotrophic, these includes chemosynthetic bacteria. That generally example was this are sulfur oxidizing bacteria. Sulfur oxidizing bacteria. And apart from the chemosynthetic, it further includes photosynthetic. And in photosynthetic, it generally includes the blue-green algae and is known as a cyanobacteria. And when we discuss about the Protista, the mode of nutrition in Protista, they are heterotrophic only, not the autotrophic. And in heterotrophic, they include the holozoic, saprobic, and parasitic mode, etc. Some Protista also act as autotrophic mode, but that are very few and known as an exception category. The next food mode we discuss in fungi. Fungi are also known as a heterotrophic mode of nutrition. That heterotrophic includes decomposers, symbionts, and parasites. And when we discuss about the plants, plants are mainly autotrophic, where some are partial heterotrophs. For example, pitcher plant. Partial heterotrophs. So, according to the discussion, the right answer is Monera, in which mode of nutrition are various as compared to fungi, Protista, and plants. So the right option for the question is option A, that is Monera, which includes various mode of nutrition as we discuss and compare with another one. Let's move on to the next question. The next question is transduction in bacteria is mediated by option A includes plasmid vectors, option B phase vectors, option C cosmids, and option D FFF vectors. So better understand, first of all we discuss about the process known as transduction. As we all know, in bacteria, the reproduction is generally by the transduction, conjugation, and the transmission. In the transduction is a recombination, sexual recombination in bacteria generally, because we are discussing about the reproductive mode of the bacteria, and in this reproduction where virus, it means phase are generally bacteriophage, act or help in transfer genetic information from a donor cell to recipient one. Donor cell to the recipient one. The process of transduction was given by the scientist named as Zinder and Lederberg. In Salmonella species, and that discovered in Salmonella species. In this process, especially in the transduction, a virus named as bacteriophage, may pick up gene of host in place of its own gene during, and that exchange generally occur during its multiplication in host cell. And such kind of virus, important thing to remember, that such kind of virus are not virulent. So after discussing the various aspects, the right option is this are: the transduction is the process in which involvement of virus So after discussing the various aspects, option B, that is the phage vectors, and phage is generally occur as a bacteriophage, and that phase are occur in the virus category. So option B is the right one in this case. Let's move on to the next question. The next one is Azotobacter and Bacillus polymyxa are the example of option A are pathogenic bacteria, B is a decomposer, C is a symbiotic nitrogen fixer, and D is a non-symbiotic nitrogen fixer. As we all know, Azotobacter Azotobacter is a saprotrophic free living bacteria that generally perform the nitrogen fixation. Important bacteria that generally help to fix out the nitrogen with the root nodules of the higher plants. So it undergo the various process and important process known as nitrogen fixation. And the next is Bacillus polymyxa. In the Bacillus species, that is also known as a free living bacteria that generally act on nitrogenous excretions and and proteins of dead bodies of organisms. So both the factor example in Azotobacter and in Bacillus species known as a polymyxa, they both act as a non-symbiotic nitrogen fixer. Because both are able to fix out the nitrogen from the atmosphere and even from the proteins present in the dead bodies of organism. So after discussing, the right answer is option D, that is a non-symbiotic nitrogen fixer. Let's move on to the next question. The next question is, what is true for cyanobacteria? As we all know, cyanobacteria is also known as a blue-green algae. And here our option A includes oxygenic with nitrogenase. Option B is oxygenic without nitrogenase. Option C is non-oxygenase with nitrogenase. And D is non-oxygenic without nitrogenase. So better understanding about this is, as we all know, cyanobacteria is also known as a blue-green algae, and that generally performing the oxygenic photosynthesis. And these cyanobacteria and blue-green algae are unicellular, colonial, or filamentous in their colony formation or in the structure, and that generally blue-green algae and the cyanobacteria possess heterocyst, and that heterocyst help in the formation or can perform the nitrogen fixation. So after discussing all this, now we got to understand that cyanobacteria are oxygenic with the nitrogenase because they are able to fix out the nitrogen. So, after discussing all this, the right option in the question is option A, that is the cyanobacteria are oxygenic with the nitrogenase because they are able to fix nitrogen, and that fix due to the presence of enzyme named as nitrogenase. Let's move on to the next question. The next question is, in bacteria, plasmid is. Option A includes extra chromosomal material, B includes main DNA, C includes non-functional DNA, and D is a repetitive gene. First of all, we discuss about what is plasmid. As we all know, plasmid is extra chromosomal DNA that are present in most of bacteria cell, most bacterial cells. Apart from that, it is a very small piece of circular DNA. The nature are generally circular in nature, not the linear DNA. It generally perform or act as a very important in bacterial life. What is the role of plasmid in the bacterial life? That generally help the bacteria to confer the resistance. First or major property is the resistance to one or more antibiotics. Due to the presence of plasmids, bacteria are resistant to the various kinds of antibiotics. Next is a desired peace. This is the first one. The next one is desired piece of DNA can be inserted, of piece of plasmid can be inserted into another one and help in the major process known as genetic engineering. So it is valuable for industrial production as well. So in industrial purpose, the various example of plasmids are human insulin. Apart from the human insulin, the next are the various growth hormones, and various antigens for vaccine. So, and that are various successful process by the including of plasmid. So the right answer for this is, in plasmids in bacterial cells are extra chromosomal DNA, and that DNA are generally circular in nature, not in the linear, and that generally help in the production of industrial production of by the process including genetic engineering, and the various important industrial productions are human insulin, growth hormone, antigens. And it also helps in the importance of bacterial life persistence because they give the resistance to one or more antibiotics. So the right answer for the question is option A, that is extra chromosomal material. So moving to the next question, that is the bacterium Clostridium botulinum that causes botulism is option A, an obligate aerobe, a facultative anaerobe, Option C includes an obligated anaerobes, and D includes a facultative aerobe. So better understanding about the questions. As we all know, Clostridium is come under the gram positive bacteria. Gram positive bacteria, and they are the obligate anaerobes that generally capable of producing the special type of spores inside their bodies and known as endospores. These individual cell of Clostridium are generally rod shaped, which is similar to their name. Some foodborne disease named as a food poisoning is caused by the Clostridium botulinum, and it is also referred as a botulism, and this is also known as a muscle paralyzing disease. Also known as a muscle paralyzing disease, it is caused. Why we name it as a muscle paralyzing disease? Because it generally causes after the ingestion of neurotoxin named as botulin and that is a very important toxin. That toxins generally block the nerve function and respiratory functions, and the next one is musculoskeletal. That toxins can be destroyed by normal cooking. So after discussing the various aspects about the botulism and what kind of toxin it is and what are the precautions, and they can die by the process of normal cooking, so the right option is option C, that generally includes the ingestion and given the description about the botulin, that is a toxin. So after the discussion, the option C is the right one. It means or defined as a obligative anaerobes, as the Clostridium botulinum is a obligative anaerobes that generally produce endospore, as we discussed. So along with that, we are done with our session. Hope student you like the session. For such more series, stay tuned with the Meso Studies. See you in the next session. Thank you so much.